Dear ladies and gentlemen, treatment and cure of all of infectious diseases and cancer depended our right knowledge, how really is working adaptive immune system in mammals, including humans. I have to say that our knowledge about how is really working adaptive immune system in human being is just wrong and not right, and therefore until today human beings do not have any other method, just vaccination. It's only vaccination, trying to treat but not cure, but treat people against infectious diseases and cancer. But this successive result is not very much. There is also many unsuccessful outcome. And therefore, now I will explain my novel knowledge about how really working adaptive immune system in mammals, including humans. My theory is new, my reverse pathway theory is new and the right one, and also I will tell about my new conception about how to work, really work in adaptive immune system. My theory and conception will bring a novel multibillion super-antibody technology for curing of all of infectious disease and cancer. As you know, it is curing method. If will be developed my theory and a new conception. My name is Victor Alexander. I am PhD in medicine, President PDPI of Capital Cell Research Incorporation, but I will try to change this name of my company to Capital Molecular Immunology Incorporation in California, United States. Contemporary clonal selection theory states that every B lymphocyte, B cells are randomly synthesizing unique immunoglobulins even before they met with antigen epitopes. But clonal selection theory couldn't explain why do immunoglobulin genes in the naive B cells undergo so-called hypermutation phenomena. It shows that contemporary clonal selection theory is wrong. Also, 3 different chromosomal location of gene exons, immunoglobulin gene exons, and different timing of rearrangements of V parts of heavy and light chain of immunoglobulin gene, bringing many unanswered questions about true molecular mechanisms of immunoglobulin genes rearrangements and synthesis of final immunoglobulin gene. Also, high antigen specificity of T cell receptors are highly questionable. The germline, contemporary germline theory and somatic mutation theory are also wrong. Let me explain my novel conception of how really is working adaptive immune system and my reverse pathway theory. First, by my new conception, development of all of B cells in bone marrow is going the same way as as pattern rearrangements. There is not going any random rearrangement of immunoglobulin gene in B cells. All of naive B cells cell development in bone marrow is finishing with the same way. There is have no randomness, as clonal selection theory says, but it is wrong. They are finishing development in bone marrow finishing the same way. There is no differences between all of naive B cells. Having of immunoglobulin M and D, IgD and IgM and IgD in their membrane surfaces. Actually, both of IgM and IgD are not often to an antigen epitope. They are not working as immunoglobulins, but both are the receptor for interaction with dendritic cell. It is my new, according to my new conception. It's anti-theory. Second, interaction with dendritic cell in lymph node or spleen. After coming from bone marrow to bloodstream and then to lymph node or spleen, they are interact with dendritic cells. They are using immunoglobulin M and immunoglobulin D to connect with the multivesicular body of dendritic cells and are getting one of the intraluminal vesicles from multivesicular body of dendritic cell. Every intraluminal vesicle of dendritic cell is full of the same antigen epitopes on major compatibility too. Because of every intraluminal vesicles has different set of antigen epitope on major compatibility too, every naive B cell is having different set of antigen epitope on, and therefore they becoming polyclonal. It is for specialists, my speech for specialists, and therefore please just take into account that it is for specialists. But if it is interested for you, please just hear until I finish my report. Third, after taking one of the intraluminal vesicles from dendritic cell, they naive B cells interact with Th4 cells, which is around lymph node, in the lymph node, many. Interaction with Th4 cells. Because of after getting a massive amount of antigen epitope on major compatibility 2 from dendritic cell, at least 50-100. Naive B cell attracts one of the local Th4 cells. After connecting of naive B cell with Th4 cells through antigen epitopes on major compatibility 2, bringing an activation of Th4 cells and begin massive cytokine production by Th4 cell. Under this cytokines, in naive B cell begins synthesis of many proteins and enzymes. One of the enzymes is unknown yet enzyme. I called it reverse ribosome, but actually it is totally different. I have to find out this after getting grant. Very unfortunately, I didn't get grant until today. Fourth, synthesis of nascent eRNA. This unknown yet reverse ribosome is using triplets of tRNAs, which is connected with activated amino acids. Activated amino acids contain amino acid plus transfer RNAs, as specialists know, which are by non-covalent affinity connection with the amino acids of antigen epitope in the endosome of naive B cells is synthesized a short 36-75 bases nascent endosomal RNA or eRNA, which is carrying exact genetic code of often amino acids to given antigen epitopes amino acids. I have called it reverse pathway mechanism or theory. 5. With the help of nuclear receptor, this eRNA relocates to VDJ section of immunoglobulin gene and replace a replaceable model. All of naive B cells is same. It is in immunoglobulin gene is same. It is heavy chain contain this replaceable model. It is PNDMP model. And only after that, V part of heavy chain of immunoglobulin gene is getting high affinity to given antigen epitope. At the same time, is going class switching to IgG constant region of heavy chain, and after that, naive B cells becoming a centroblast and is undergoing 9 cell divisions and becoming centrocytes, and they move to damaged with infection diseases or cancer areas and becoming high often IgG producing plasma cells to given antigen epitope, and some memories, and of course some memory cells. Only V part of heavy chain has affinity to given antigen epitope, not light chain of immunoglobulin G, but not light chain. Light chain is a helper chain which is connected with surrounding molecules of antigen epitope non-specifically. Only heavy chain has affinity or specificity to given antigen epitope. 6. Th and Tc cells, it is T cells, only recognize self tissues and organs antigens, but all foreign receptors recognized by T cells as a non-self antigens. All T cells do not have specificities to any foreign antigen. I am addressing to all of biotech companies of the world to support my advanced novel conception and reverse pathway research project, which will bring novel multibillion sub-technology for curing of all of infectious disease and cancer. Thank you. I can show it on slides, but very unfortunately I do not have enough time to do it. I will do it in one of the World Congresses, and it is very much explain what I am saying. Thank you.